If you're trying to understand biology at the cutting edge, you really need to know about spatial transcriptomics, or ST. This technology was named Method of the Year 2020. And for very good reason. It's fundamentally changed how we study cells.
It really has. It gets at one of biology's oldest problems, context. Traditional RNA sequencing requires you to take a piece of tissue, grind it all up, and then just look at the average gene expression.
So you get a perfect list of ingredients, but you have no idea what the recipe was.
Exactly. You lose the recipe, the kitchen layout, everything. Spatial transcriptomic, on the other hand, it keeps the cell's address, its spatial context, while it quantifies the gene expression. We can finally see who is expressing what, and crucially, where they are.
Okay, so we've moved from analyzing, the average traffic flow in a city to suddenly having the GPS coordinates and maybe even the job title of every single driver. That is powerful.
It is. But even with these amazing tools like Visium or CosmX, there's huge bottleneck.
But data sparsity.
Data sparsity. Precisely. A lot of current ST tech just has low sensitivity, or it's designed to only look for a limited set of pre-selected genes.
So there are just huge gaps in the data.
Huge gaps. And if a really critical gene for, say, a disease isn't on that pre-selected panel, or if its signal is just too weak to pick up, we miss it completely. We're left with this sparse, really noisy data.
Which is where imputation methods come in, right? These computational tools that are supposed to fill in the blanks. We've seen some before, like KNN methods or deep learning tools like Tangram. So why didn't those fully solve the problem?
Well, their core failing, really, was focusing on predicting individual genes. And if you think about it, individual gene expression is just inherently noisy. It's variable, even a perfect sample. So trying to guess the exact level of one missing gene, it's an incredibly unstable task.
And the results end up being just as noisy.
Exactly. And a lot of those methods also tend to overestimate spatial smoothness. They just blur everything together, wiping out real biological boundaries, like the sharp line between a tumor cell and an immune cell.
So the solution required a conceptual shift. We need to stop focusing on the individual notes and start listening to the whole symphony.
That's a great way to put it.
And that brings us to the innovation at the heart of our deep dive today, PASTA, which stands for, and this is a mouthful, Pathway-oriented Spatial Gene Imputation.
Right. So PASTA doesn't try to predict single noisy genes. It predicts the expression of entire biological pathways.
And that's the critical difference.
It is. A pathway is a functional unit, a set of genes that we know work together to do something like, say, drive inflammation or cell growth. By aggregating the signals from all those genes together, PASTA naturally smooths out that high individual gene noise. It combines that with cell type information and spatial proximity and gives us something much more robust and biologically meaningful.
So our mission today is to really get under the hood of PASTA, understand how it pieces all this information together, and then look at the hard evidence from these compelling case studies in cancer and neuroscience that show why this pathway-level approach is just better.
Okay, let's turn to the design, the mechanism itself. The shift to pathways aggregating signals, that feels like the first really big idea here. Yeah. Why does that aggregation give us cleaner data, just right off the bat?
Let's stick with your orchestra analogy. It's perfect for this. A single gene is one instrument. Let's say one musician takes a breath or drops their sheet music for a second. That one note is missing. That's your technical noise, your sparsity.
Okay.
But the pathway. The pathway is the entire brass section playing a chord. So if one trumpet player misses their note, the overall sound of that brass chord is still loud and clear.
Because the other trumpets, the trombones, they're all still playing their part in that coordinated function.
Precisely. That functional aggregation just drastically improves the stability and the biological relevance of the signal. So to make this work, PASTA needs a few things. What are the inputs?
It needs three key inputs. First, the raw, sparse spatial data from the tissue you're studying. Second, a very detailed reference map of gene expression, which usually comes from single-cell RNA sequencing, or scRNA-seq.
And third, it needs a list of all the genes that belong to the biological pathways we actually want to look at.
Exactly. With those three things, the core computational challenge is to build what's called an alignment map. It's a matrix, really, that connects every single cell from that high-detail reference onto the actual physical spots in the sparse spatial data.
And how does it build that map? What's the logic?
It guides the mapping process using two foundational assumptions. First, that cells of the same type should, have similar expression patterns. And second, that cells that are physically close to each other should also have similar expression patterns.
But hold on, if the two data sets, the spatial data and that single-cell reference, were generated on completely different platforms, how do you even begin to align them accurately? That sounds incredibly difficult.
It is. And that's where the elegance of PASTA's training process really comes in. To solve that exact problem, PASTA minimizes a weighted sum of 4 specific training checks. You can think of them as loss functions.
Okay, so let's call them 4 specialized quality control coaches, each one guiding the model during training.
That's a great analogy. 4 coaches, each focused on a specific performance metric.
So let's meet the first coach, the reconstruction check.
Okay, so this coach is the local cell type specific checker. It looks at the predicted pathway expression and compares it to what was actually measured in the spatial data. But, and this is the crucial part, it only demands similarity within a very specific context, the surrounding cells that belong to the same cell type.
Ah, so it forces the expression to be smooth within a population of, say, immune cells, but it allows for a sharp, realistic jump when you cross a boundary into a different population, like tumor cells.
You got it. It prevents the model from just blurring everything together. It respects biological boundaries.
Very clever. Okay, coach number 2, the global similarity check.
This one handles the big picture. Since we are merging data from 2 totally different technologies, this check makes sure that the overall transcriptome profile of the mapped single-cell data broadly matches the spatial data. It's a general alignment across all the genes that the 2 datasets share. It just maintains global coherence.
And then the third coach, which you mentioned, is maybe the most critical piece of PASTA's innovation, the pathway similarity check.
Yes, this is PASTA's focusing mechanism. So if the global check looks at all shared genes, this pathway check narrows the focus to look only at the genes in the specific pathway we're trying to predict.
And why is it so important?
Because highly expressed genes that are not part of your pathway, common housekeeping genes, for instance, can easily drown out the more subtle signal of the pathway itself. By focusing the comparison so narrowly on just the pathway genes, PASTA ensures that final alignment map is perfectly optimized for the biological function we actually care about.
I see. And finally, the fourth coach, the neighborhood similarity check.
This is one more layer of spatial quality control. It encourages the model to produce these cell-type coherent expression patterns by looking at how similar the pathway expression is in the immediate surrounding cellular neighborhood.
So all 4 coaches work together.
Exactly. Combined, they ensure the final predicted pathway expression is accurate, stable, and biologically informed because it's blending cell identity with physical location.
So to recap this first part, PASTA's architecture is a huge leap forward because it imputes pathways, not single genes, which boosts stability. And it uses this really fine-tuned training process with 4 checks that simultaneously leverage both cell type identity and spatial proximity.
Okay, so now the big question is, does this clever design actually translate into better results when you're facing the messy reality of biological data? The researchers put PASTA through some really rigorous stress tests using simulated datasets to find out.
So let's start with the baseline. When everything is pretty clean, how did it do?
In a straightforward scenario, PASTA just instantly showed superior predictive accuracy. It achieved the highest correlations and the lowest overall error rate compared to all the established methods like Tangram, Surratt, and Leisure.
So right out of the gate, even with clean data, the pathway approach is just better.
It is. It confirms the core idea. But you're right, that's the easy part. The real stress test comes when the data is more realistically challenging.
Like the partially observed pathway setting, where you can only see a subset of the critical genes you need.
Right. And as you'd expect, performance declined across all the methods when information was missing. But PASTA still maintained its superior lead. It showed this incredible robustness, which really demonstrates that its pathway-based architecture is just less brittle. It handles missing information far better than methods that are totally reliant on single genes.
They then took the stress test even further by deliberately mismatching the data sources. So this is like trying to build a map of a person's tumor, but your reference cell atlas came from a totally different patient, or maybe even a slightly different part of the tissue. How did PASTA handle that kind of fundamental variability?
They simulated 3 really tough conditions. First, they used data from the same subject but different tissue regions, which means the cell populations are going to vary a bit. PASTA remained notably stable.
Okay.
Then they simulated a case where the single-cell reference came from a different individual entirely. Even with that massive variability, PASTA still came out on top, showing superior performance and stability compared to its competitors.
Wow. And what about the hardest test? What if the reference dataset didn't even have the full range of cell types that were present in the spatial data you're trying to analyze?
That's the real challenge. And methods like SIROT, which are highly sensitive to cell type differences, they struggled significantly. PASTA, of course, saw a natural decline in performance, but it still provided satisfactory and scalable predictions.
So its dual focus on spatial context and pathway function allows it to sort of bridge the gaps when cell types are missing.
It does. It can handle that uncertainty. It's also really interesting to note they found that just having more reference cells isn't always better. An excessively detailed, or what they call over-clustered reference map actually hurt performance because it introduced all this unnecessary micro-heterogeneity.
So smart data prep is key, not just sheer volume.
That is a crucial takeaway for anyone working in this field. PASTA's robust performance across all this mismatched data, different regions, different subjects, missing genes, it just proves its pathway focus is a powerful antidote to the noise and variability you always find in real-world biological data.
So let's move from the computational metrics to the actual biology. The ultimate measure of success has to be whether PASTA provides new insights or validates known biology with much greater clarity. Let's start with the case study on human lung cancer tissue.
Right, so they analyzed 44 different cancer-related hallmark pathways.
Yeah.
The results were, well, they were astounding. If you take the hedgehog signaling pathway, for example, which is vital for regulating cancer stem cells.
A really important one.
A really important one. PASTA achieved a correlation score as high as 0.907 with the known ground truth.
0.907. I mean, in biological data, that is a fantastic level of accuracy.
It's incredible. And the question is, did these high correlation predictions actually translate into the correct biological mapping?
And did they?
Absolutely. PASTA accurately localized key pathways where they were supposed to be. For instance, pathways associated with tumor cell proliferation, like MYC targets V1 and V2, were correctly enriched right in the tumor cells, specifically in the basal cell population.
And what about the immune response?
Conversely, immune-related functions, like the inflammatory response, were correctly highlighted in the immune cells, in the T cells and macrophages. And the source material really emphasizes that older methods, the ones that rely only on measured genes, they often fail to achieve this kind of precise enrichment mapping across these distinct cell types.
So the functional information is much sharper. That must allow for better structural analysis, right? How did they visualize the tumor structure using this pathway data?
They used a clustering tool called IRIS, which is designed to spatially segment tissue into meaningful domains. And when they clustered the tissue using PASTA's imputed pathways instead of noisy single genes, they got a much, much finer separation of these domains.
So what does that mean in practice?
For instance, PASTA could successfully distinguish between tumor cells that were located inside a dense infiltration of immune cells versus those in the main tumor core. That's a highly nuanced distinction that is essential for understanding the tumor microenvironment.
And this clearer structure also helped with temporal analysis, I believe. They did something called trajectory inference using Monocle 3. Can you explain what that is?
Of course. Monocle 3 is an established software that uses gene expression patterns to estimate the developmental or disease progression sequence that cells follow. It's called pseudotime.
So it's like creating a timeline for the cells.
Exactly. And because the PASTA pathway data is so much less noisy and computationally lighter than thousands of single genes, the trajectory analysis was much cleaner. It correctly mapped the progression. The most developed tumor cells were concentrated right in the center of the tumor domain.
Which aligns perfectly with cancer theory.
Perfectly. And it was validated by the physical distribution of the ME gene, which is known for driving abnormal cell growth.
Let's pivot to neuroscience. Case study 2, mouse brain development. This whole field is really dominated by the concept of inside-out neurogenesis.
That's the classic rule book for how the cortex develops. The brain generates the deeper layers, layers 5 and 6 first, and then it sequentially builds the more superficial layers, layers 4, 3, and 2, right on top of them. It's a very precise temporal and spatial roadmap.
And did PASTA manage to correctly infer this established developmental timing?
It did, with remarkable accuracy. Using what are called GOBP pathways, these are standardized lists of genes involved in processes like neurogenesis, PASTA inferred a cell pseudotime that perfectly matched that inside-out pattern. L6 cells showed the earliest pseudotime, confirming they were generated first.
And how did that compare to other methods?
This is crucial. The authors specifically note that the predictions generated by a competitor method, Tangram, failed to capture this specific established biological time shift.
Wow. And finally, a quick look at their stability metric on human frontal cortex data. They use something called the tissue calibration score. What does that measure?
The tissue calibration score is basically a metric that assesses the uncertainty and the stability of the imputation itself. A lower score means the model is more confident and the estimation is more reliable.
And PASTA's score was?
In this brain data, PASTA achieved a significantly lower median calibration score across all the cell subsets compared to competitors. So this doesn't just mean PASTA's results were accurate, it means they were more trustworthy and more stable.
This has been a fascinating deep dive. It's clear that PASTA successfully addresses the Achilles heel of spatial transcriptomics, that data sparsity, by making this really critical shift from focusing on noisy individual genes to robust, biologically meaningful pathways. By integrating spatial location with cell type identity, it just delivers highly accurate and stable insights across these incredibly diverse and complex tissues.
And while the results are excellent, the authors are very clear about the remaining limitations. Right? PASTA is heavily reliant on the quality and completeness of that reference's scRNA-seq data. If your reference is missing key cell types or is just low quality, the imputation is going to be flawed.
Garbage in, garbage out.
It's always true. And as we mentioned earlier, pathway selection is still critical. You have to make sure the pathways you're targeting are actually relevant to the disease or the tissue you're studying.
And I imagine in some extremely chaotic microenvironments, the assumption that cells nearby will have similar pathways might start to break down.
It might. And that's a challenge for future refinements. But looking ahead, the real trajectory here is integrating even more layers of data, maybe proteomics or metabolomics, alongside PASTA's pathway approach. The foundational success is translating these spatial maps directly into functional understanding. We're not just seeing where a tumor cell is, but how its signaling is active.
We've seen PASTA validate the inside-out neurogenesis model and precisely map cancer signaling in the tumor microenvironment. So considering how effectively pathway imputation can clarify these highly complex dynamics, what uncharted biological process in a challenging tissue, maybe in a chronic inflammatory disease or a complex organ like the liver, do you think needs this kind of reliable pathway-level spatial mapping next? Let us know what you think. That's all the time we have for this deep dive.